Hello, everyone. Welcome to Lab 1. So in Lab 1, Histology section, we are going to be going through two different labs on this. And the first one, I think, is usually the first overwhelming because of how similar things look. So it'll be my job to help you guys try to visualize these things better. And then also what we've got is some things to help you through to study it and prepare for that first exam. The exam on Lab 1, Histology section, takes place on the third week of the semester between August the 31st, and September 4th, between that time frame. And that's when you will see your tissues exam. It will be 30 multiple choice questions and 20 identifications. They will randomize in order, so it won't be in that order, but you can go back through that exam. And I always like to start off kind of what is your exam going to be like so you can kind of be thinking about it. Now, in the examination, what I've done to help you also prep for that examination is if you also click into D2L, what you'll see is under content, you'll see not only my lab PowerPoint, but some flashcards that I made with some basic instructions on how to make it work. You print these off following the instructions. If you actually go in, print them off 2 per page. How do you do that? Well, hit File, Print, enable printing, file print. Now, what you have to do is instead of print full page slides, you want to do this. Okay. So when you print it like this, it will make on a sheet like this. Now you can choose if you want to do it landscape or a portrait, whichever one works best for you. It really doesn't matter. And you cut this in half. And then what you're going to do after you cut this in half, just fold this over so that the picture faces one side and the name, locations, and functions are on the other side. What you can then do is you can write in and fill these out based on the pictures before you fold them. Then fold them up, and then you'll look and see the picture and be like, okay, this is this tissue. This is stratified squamous. It's located in the skin. It functions in protection, okay? And da-da-da-da-da. You could put all that in there. And then as you go through, I randomize them, but these are the exact pictures from the test. Now, they are found in my notes, so you can go through my notes and find the answers, but you can fill those out by looking through this, but they are in a randomized order on purpose. But it walks you through a basic instruction on how to make those, and those same identical pictures are the ones used for the exam. Now, what we're going to start off with is here's a list of things that we are expected to cover through Chapter 4. And I do try to test on everything that I possibly can. Now, it is not everything's not in there. It's really not something that I can test on everything, but I try anyway on here. Now, we want to start off to see what tissues are. Now, I want to do something just to kind of help you guys, and this is something you would have seen in a lecture. If it's not in my notes, you are not going to get tested on it. If it's not in the structure sheets, you're not going to get tested on it. What I'm more so doing is just talking about something that you might have seen in your lectures already. Now, in lecture, let me get, come on, it says to focus. Focus. It still won't do it. Why is it? I tell you. This blasted thing. It's I unhook everything because I needed to take it somewhere, and now it's not going to work for me. There we go. Okay, so let's say, if we will, let's just change that a little bit. I want to see more of the paper. Okay, less of the camera. Okay. So let's say, guys, that I had a pyramid, and I'm looking at one part of the pyramid. We would have very small things up here, things like subatomic particles. I would have atoms. I would have molecules, organelles, cells, tissues, organs, organ systems, and then an organism, you. Okay? And this is kind of what we would see in a human, is that we are built up of these really small things that gets to the really big thing, you. And it's right here is where we are in lab, is tissues. Tissues are composed of cells and make up organs. So, for example, if I had a liver, for example, that I took out of your body, and I removed it, and I took a little knife to this, and my little knife were to cut open the liver and take a piece of the liver out, what we might see is if I have a little chunk of liver here that I've removed, I could then take a look at the tissues by putting it on a microscope slide here and put a little piece of that liver on it and look at it, okay? So that's what we're basically doing is we're taking a sample of an organ, okay? A sample of it, putting it on a slide, and we could see the cells that make it up. So what's interesting is, guys, you have a sample of an organ that contains tissues, but there will be more than one tissue. And that's a big point I want to make, okay? Because that will help you understand what you're seeing better than anything I think I can say. Now, tissues, they are a collection of similar cells and their products. They work together. They can create organs, okay? And the study of this is called histology. Histology is the study of tissues. Okay, histology. Think about a tissue when you have histamines. You need to blow your nose on a tissue. Histology, the study of tissues. Now, as you can see, tissues are composed of cells, and that tissues makes up organs, okay? And when we look at that, the tissues of the body here, they are going to come together to produce our organs of our body. And one organ may have multiple different types of tissues in them in a unique combination. Okay. Now, case in point with that, there will be 4 different types of tissues in the body. There's epithelial tissues, which we have to know 8 of those. There will be connective tissues, which we have to know 12 of. 3 types of muscles that we learn, muscular tissues, and 1 type of nervous tissue that we have to learn. Now, the numbers of them are not as important because that is, of course, things that we're learning. We said these are types of tissues in the body, and these tissues have these types. But what I want you guys to be thinking about more than anything is these are the 4 major groups of tissues that exist in the body: epithelial, connective, muscular, nervous. Now, as we go through that, we want to see what epithelial tissues actually are. Now, I want you guys to kind of imagine the table somewhere near you. There might be a table. And on that table, there might be a top. So if we take a look at the table I'm sitting at right now, I'm sitting at a table in my home office, and this table here has a top. It's lined here. And this is covering the outer surface of that table. And then under it is another surface. And these 2 surfaces cover the inner and outer surfaces of this table, so this has a complete lining. And just like that, my body here, I have inner and outer surface coverings of my body. And that's what my epithelial tissues are. They are what covers me inside and out, internal or external surfaces of the body. Okay, this covers your inside and outside. So the lining of your mouth, your skin, the insides of your eyelids and the outsides of your eyelids, the inside of your anus and the outside of your anus. Basically, we have this continual layer of epithelial that goes inside and out that basically covers us completely internally and externally. Then there are glands that are also epithelia. The epithelia have only, besides that, but they have glands. And glands are something that makes secretions. They make fluid secretions. Now, later on today, we're going to talk about the different types of glands and types of secretions. So we know the epithelia and glands both make up the epithelia together. Now, the epithelia proper, we might say, are the things that cover us inside and out, but also the glands of the body. Now, as we go through this, we said that epithelia covers us inside and out. So given that, it acts as a physical protector. For example, from abrasion. Okay, let's say I rub up against a brick wall a little bit. Well, that's going to prevent my arteries from being abraded so easily so I don't nick a major artery and bleed to death. Or prevent me from dehydrating as easily. Now, we do get dehydrated if we don't drink enough, but the skin is a little bit waterproof, not completely, but not perfectly, but partially, and it does resist water loss, but also can prevent chemical or biological agents from getting in. It acts to act as a block to prevent certain viruses and bacteria and fungi and things like that from getting into the body. But beyond that, also some chemicals. Now, some chemicals can absorb through the skin, like nicotine or other things. That's why we give transdermal patches for people who want to quit smoking. We can control the permeability, for example, absorption of the body, absorbing nutrients and things like that. They can provide sensation. Most epithelia have a very large nerve supply. They really will be connected to lots of nervous tissue. And that's because epithelia that's on the outside, specifically in the nervous system, come from the same area of development. So we link them together. And most specialized secretions out there they produce here, things are glandular epithelia, for example. Their job is to make secretions like saliva or sweat or body oil or hormones or digestive juices, enzymes, acids, stomach juice, right? Things like that. So as we go through there, now two cells have to be added together. So when I make a tissue, for example, I have a cell here and this cell. And this cell have to be able to attach to each other somehow. And there are a variety of ways to make them attach. One way I can make them attach is go here at the very top. And at the very top, I can use a very tight knit bit of proteins that come together here. And we can refer to those as tight junctions. And a tight junction, what they act to do is they are waterproof. They are found at the top of a tissue, usually like in the kidney or something. Then we can have another one here that is often found at the bottom. Here, that might help attach this tissue to the bottom layer. And also sometimes found like this as well to attach each other. And these are called anchoring junctions or referred to as desmosomes. Anchoring junctions or desmosomes. And then also I can have one where it passes through, but it's a hollow tube to let stuff pass through, like water molecules, for example, might be able to pass through here. Ions might be able to pass through. Small chemicals can pass through, okay? And larger chemicals can pass through. And these are referred to, guys, as gap junctions. Now, you do need to know these are the three types of intercellular junctions, okay? Intercellular junctions. There are three types of intercellular junctions: tight junctions, anchoring junctions, and gap junctions. And one of the things we'll see, we call tight junctions occluding junctions. And we call macula adherens, those are anchoring junctions, or desmosomes. What I'm going to do here is call them desmosomes. I sometimes refer to them as anchoring junctions because that's what their job is. But I always sometimes forget, like the book will refer to them as macula adherens. anchoring junction, adhesion junctions. They are proteins mostly made of what's called cell adhesion molecules, things like that, that help to hold two cells together. A cell adhesion molecule is a very interesting little protein. I sometimes worry that my cell adhesion molecules will somehow give up the ghost and they'll all let go at one time, and I'll just be a puddle of goo. And I don't want that to happen. That always worries me. So, as we think about these three types of intercellular connections or intercellular junctions, cell junctions. Now, also, we have what is called a basement membrane. Now, as we talk about basement membrane here, what I kind of want to do is I'll sketch a little bit here, is that I have this thing that is kind of here like that. I'm going to put a dotted line in the middle. And then what I'm going to do is on top of that, I'm going to put some cells. And let's say I've got some cells that are sitting on top of this, and some epithelial cells of some type are up here. And there's some little nucleuses that they have. And let's say I've got my epithelia right here. Okay. This is my epithelia. And under this, there is going to be a thing here called a basement membrane. And the basement membrane has actually got two layers to it. There is what's called for the basement membrane, there is two layers, a basal lamina and a reticular lamina. Now, the basal lamina and the reticular lamina. Now, underneath the reticular lamina, we will find various connective tissues under here. Okay, so connective tissues are deep to that. So what I'm trying to show you is that the basal lamina is actually made by your epithelium, so it's close to the epithelium, and the reticular lamina, the deepest layer, is secreted by the connective tissues. And there, where the basal lamina is, the basal lamina has two subdivisions to it. What's called the lamina lucida and lamina densa, or the clear and dense layer. Think about it. If you're thinking clearly, you're lucid. If you're very dense, densa. Densa is the Latin word meaning dense. Lucida is clear, Latin clear. So if you're lucid, you're thinking clearly. Okay, the clear layer is called the lamina lucida. Now, or lucida. So what we want to make sure we see that those things make up that. Now, I do like to talk about that epithelia. There is a basement membrane at the bottom and an apical surface at the top that I do want to talk about. Now, we don't get too much into that, but I at least wanted to have that on there. Now, another big thing that I want to talk about with epithelia is there's two ways we can classify them and name them. And we can name them based on two factors, okay? Based on the shape of the cells at the top of the tissue, the apical surface, or on the number of layers. Now, as we think about shapes, let's talk shapes first. There is 1, 2, 3 shapes. Now, the first shape we're going to see, this first shape that we're going to notice is going to be very flattened. Okay, very flattened here, like a little pirate hat. And it's going to look like that from the side. And if we look at it from the top, we get something that looks a little like a fried egg. Okay? And this little fried egg appearance, the word that's used to describe this shape is referred to as squamous. Squamous. Squished, squashed, squamous. Think of a fried egg. Fried egg in a skillet. On the side, you will see it looks very different than it does on the top. Now, there is another one that if I looked at it from the side, it would look like this, fairly boxy. Fairly boxy. You're going to see pretty much your nucleus here is going to pretty much be the same distance from all surfaces. Okay, should be roughly the same distance from all surfaces. And if you look at it from the top even, it's going to look the same way. You won't even really be able to distinguish much of a difference here. And this side here, it looks boxy. This one is, or a cube, so it's called cuboid or cuboidal. Now, if we also were to look at one, there is a type of tissue from the side that's long and tall. And it's long and tall, but if you look at it from the side, you will also see it pretty much looks like this on the sides. But because it's long and tall, it looks like a columnar. So we have squamous, cuboidal, and columnar, like a column. Now, so this guy here being tall, okay? And so what this tells me is that we need to look at the sides of the tissue from the side. Now, that is by shapes. Number of layers is the other way that I can describe epithelia is based on their number of layers, not just the shape of the cells at the top of the tissue, but their number of layers. Now, for example, let's say here I have my basement membrane. That's the basement membrane. And that basement membrane made up of a basal lamina and reticular lamina. Let's say that we have cells here. And this is me trying to draw some of these fried eggs. Okay. And there's only one layer. You notice that there is only one layer. We call this simple. If there's two or more, so let's do this, and then let's go in and draw this. Let's draw one layer. And draw another layer. And I always like to put a nucleus in each one of my cells just because, you know, a cell ought to have a nucleus. Am I right? Now, what you're going to see here is, guys, this is 1, 2 layers. We call this stratified. Now, based on the shape of the top, you can look at a cell and say it's stratified squamous, or it's simple cuboidal, or it's stratified columnar, or simple columnar. So you can have simple squamous, simple cuboidal, simple columnar. You can have stratified squamous, stratified cuboidal, and stratified columnar, based on the shape of the cells and the number of layers. Now, we usually put simple and stratified first, and then we actually put the shape of the cells at the top second in the name. So, that's how we go and do that, okay? So you can have simple and you can have stratified, okay? How we classified them. Now, that's a very important thing to help you guys understand it. Now, then what we're going to look at is, guys, is let's say we have cells at the bottom. Let's say that my skin is now dried, dead, and flaky, and some of it flakes off. Those are dead skin cells. Well, if my dead skin does not replace itself, eventually I'm going to run out of skin. But thankfully, my epithelium is always replaced through continual division of what is called basal cells. Basal cells are stem cells called germinative cells. They are found near the basal lamina. They are the ones that divide. We call them basal cells. This is the cells, for example, in the skin, the basal cells of the skin is where you get basal cell carcinoma. So as we go through this, we will see epithelial tissues. Now, epithelial tissues, there is such thing. So here, as I said, we have a simple and a stratified of all three shapes. So there is simple squamous, there is simple cuboidal, there is simple columnar. There is stratified squamous, stratified cuboidal, stratified columnar. But there are two that don't fit this. They're called transitional epithelium and pseudostratified ciliated columnar epithelium. Now that is the word that's the first word that students hit in A&P going, holy cow, have I got to spell that? Yeah, you will. It's always on my test. It's my favorite tissue because I just love saying pseudostratified ciliated columnar epithelium. It's like wacky inflatable arm flailing tube men. It's just fun to say. Pseudostratified ciliated columnar epithelium. And so as we go through it, I hope you guys find it that if you practice these things, you'll need to write these words out because it is the tissues that we have to identify. Now, here's what I want you guys to be doing. Now, first things first. When you guys go, and if you look at a slide, first look at the slide and see is there one or two or more layers. Then, so first thing you're gonna do is you ask yourself, is it simple or is it stratified? Okay, first things first, simple or stratified. That's your first thing. Then secondly, look at the top layer. If it's stratified or simple, whatever's at the top will be what's at the top. If it's simple, it's only one layer. Look at the shape of the cells at the top. So this right here would be stratified squamous. This would be simple squamous. Squish, squash, squamous, or squamous. Now, so that's what we do. Now, also epithelia, because epithelia, they are what is exposed to the air. They are always exposed to a surface. Epithelia, because they line the surfaces of the inside or outside the body, they must have a free exposed surface. So look for a free surface. I'm going to show you that in every slide that I give you guys. That is like my thing you want to look for. The telltale sign of an epithelium is a free surface. There is somewhere that it is exposed to the air, whether it is inside of a intestine, on the insides of the gut, or on the outer surface of the skin, or the lining of the mouth. It will always be exposed to some environment. Always, always, always. Okay. So you look for that and you go, oh, I see a free surface. It's an epithelium. Now that I know it's an epithelium, is it simple or is it stratified? Then what's top shape of the cells at the top? And you will identify it. That is the simplest, easiest checklist of things to go through in your mind to get you guys through this. Now, for some reason, this picture here ended up here. And I don't know what was going down. But normally what I do, but I can't edit it here, and I've edited it, but it's just annoying. I don't know why it was on there. I must have been moving something around. I go through and edit my notes on a frequent basis, just trying to find things that need to be here, there, and everywhere, need to be fixed or neat or nice. Now, what I wanted to look at is that, now, one of the things, the descriptions are not something I'm going to test you on, but they are there to help you. Now, it's going to be a single layer of oval flattened cells, looking like a bunch of fried eggs, but remember on the side what you're looking at, right here. Now, here it is on the side. The arrows are pointing to it. This thin layer right here, very thin. This is the lining of an artery or a smaller blood vessel. Here, artery-like, an arteriole maybe, and here we have a smaller vein. And we're in a muscle, the skeletal muscle of the body. And you can see here that these are my little cells that line the blood vessels, my simple squamous. And each of these arrows are pointing at a simple squamous. So it's only one layer. Now this other stuff here, that's smooth muscle tissue. Here's some connective tissues. Here's skeletal muscle tissue. And you're like, how do you know? I've been doing this for a very long time. Since I was an undergrad, I had to be able to look at this stuff and know what I was seeing. These are blood vessels found inside of a muscle. So somebody took a muscle and cut it open. These are bits of muscles. And so what you're going to notice is one of the things on my test I'm going to do is have a picture, and I'm going to say, name this tissue. Um, and what I normally want is this slide here to be right here, is where it's supposed to be, is right here. And then this is supposed to be gone, but this is what it's supposed to look like. So I think it looks like fried eggs when you look at it. Now, we find it lining blood vessels, lymph vessels, and body cavities. It also covers partially things like your liver, your stomach, your gallbladder, and other visceral organs. And it also is found in the kidney. Now, I don't think it says in our lab manual, structure sheets, nephron, the loop of Henle. Nephron loops in the kidneys. You're like, what's a nephron loop? You don't know what that is. I would say it's in the kidneys, and if you said it's in the kidneys, hallelujah. That's one of the things I do in my slides. So on my test, here's one of the things I'll be doing. There will be a picture like this right here that will be on the test. It'll come up and it'll say, for this tissue, give me the name of the tissue, one of its locations, and one of its functions. So of the many locations and functions I gave, like it's in the lungs, here you can find that it's in the lungs. It allows for diffusion. This right here, lungs, can count as a location if you like. I don't like the way that our structure sheets outline this. I've been saying for some time we really need to change these structure sheets for the tissues. All right, and so anyway, the simple cuboidal epithelium. So now that we've seen simple squamous and we understand why it's simple squamous, simple cuboidal, right here we have the cells, and you can see that these cells are very boxy shaped, and there's only one layer. Here we have connective tissues underneath that. And there are a single layer of cube-shaped cells there. They should be equal in height. Kidneys and glands, simple cuboidal. Think of kibbles and bits, kidneys and glands. And what do they do here? They secrete and absorb. They secrete and they absorb. That's what these two things do. Great. Is they secrete and they absorb stuff. Okay, we're actually going to talk about this, how these cells do that in the kidneys. We'll talk about that they do what's called secretion and absorption in the kidney, and they're in kidneys and glands. So you can see why it's simple right here, especially one layer of boxy-shaped cells. And then all this stuff here is connective tissue. So these are nuclei of connective tissue cells found on the outside. But see this line right here. This line is the basement membrane. And this is your free space, the tube opening where the urine to be that the kidney is making would actually be collected. It's going to be located here. It's where the fluids that's going to become urine end up being inside. And the outside here is used to produce these tubes are used to make urine. That's what their job is. Kidney tubule. Secretion and absorption. And it also makes your, like, for example, your kidneys, but it also secretes in glands. This is most of your cells that make hormones or that. Simple columnar epithelium. Simple columnar epithelium, as you can see here, one layer of long and tall cells. There's one cell. Here's one cell, here's one cell, there's a cell, here's a cell, long and tall, one layer, okay? Now, these cells here, they usually have a nucleus kind of located a little bit further down near the bottom, usually. They may possess mucus-secreting cells called goblet cells. Think of a champagne goblet full of mucus. So think of that mucus goblet. Goblet cells make mucus. Now, this tissue lines your digestive system. It's found like in your uterus and your stomach. Now, I'm going to mention it, but I will not test on it. I'm not going to test on intermittently ciliated simple columnar epithelium. I think that's a little bit too detailed to test, but there is a specialized tissue in the uterine tubes called intermittently ciliated simple columnar epithelium that the cilia will wave, and it will help move the ovulated oocyte or egg to the uterus for pregnancy. Now, a thing that I do want you guys to understand pretty well is what's going on the top of the cell. There is a thing that the cell may have on top. And let's just basically say that we want to go on vacation. You're going on a family vacation. It's summertime. You finally can go on vacation. And basically, you're driving from point A to point B. Now, really, when you go on vacation, there's two fathers out there. Okay. Now, the top one here, this is going to be my dad on a trip. Okay. My dad is like, he said, boy, we got a long way to go and a short time to get there. If you got to go, there's a jug in the back. Pee in that. We're not stopping. And just goes. Now, that's one father, shortest distance. Now, the other father, that would be Clark Griswold. Holiday Road, Holiday Road, Now, if this red line was the cell membrane, would you agree that this one covers more surface area? Though the span between A and B is the same, this one covers more surface area. If this was a microvilli, that would increase the surface area of a cell so it could absorb nutrients, okay? And that's what it's doing. It's increasing the surface area for the absorption of nutrients. And that's, you can see, I mean, there's tons of them here. And look at them, little hair-like structures. And they're little fingers, microvilli, that increase the surface area that's available for absorption. So these guys secrete digestive fluids and they absorb nutrients. Secretion and absorption. Secretion and absorption. Look back here. What is it that epithelial tissues do? Secrete and absorb, secrete and absorb, secrete and absorb. Protection, secretion, absorption. Secrete, absorb, secrete, absorb. Almost all of them do those two things. So it's pretty easy for epithelia to know what they do. Okay. And I see columns when I just look here. I like to put little visual analogies in there. Now we've gotten the simple done. Let's do stratified. Now, stratified squamous to me looks like a stack of pancakes. I've got a whole bunch of pancakes all packed up together. So somebody wants some pancakes. Now, this is a slide of that, and you can see very flattened cells at top, and at the bottom the cells are boxy. Flat at the top, boxy at the bottom. And I will show the real slide. You see that there's multiple layers, and the top is flat. So it's stratified squamous. Now again, as I said, I see a free surface. Here's the basement membrane, okay? Always in epithelia, remember free surface. Free surface. You always will see a free surface, okay? Now the outer skin is stratified squamous. Your mouth lining is stratified squamous. The throat, the vagina, and even the anus, parts of the anus called the anal canal, is stratified squamous or squamous. Now, the skin, though, what's interesting is your skin surface, those outer cells are what we call keratinized. But in the rest of the body, this doesn't happen. Like in the vagina, the throat, anal canal, they remain moist. I know that your favorite word in the world is moist, but they stay moist. They actually maintain what's called a mucosa. Whereas in the skin, they keratinize. So we get what's called flaky skin. We get the layers where it becomes keratinized. Well, here it's non-keratinized inside the mouth. And it's a good physical protector. Protects. It protects. Really good at protection. Now, stratified cuboidal. Stratified cuboidal epithelium. What does it do, guys? Well, here we have to have 2 or 3 layers, at least 2, if not 3, of cuboid cells. Now, if we look at it, here's our free surface, the so-called lumen. 1, 2. 1, 2. duct. And you can see here they're very square-shaped. It doesn't look so good here on this slide as it will here, but I use the real thing. So they're in the open areas called a lumen, the open areas of a duct called a lumen. A lumen is where light can pass through. So it's like, think about a lumen, like illuminate or illuminati. When you illuminate something, you're putting light on it, shedding light on it. Light passes through a lumen. A lumen. And this is for protection. Mostly functions to protect. For example, let's say that this is a sweat gland, and the salts of the sweat gland may damage some of the other tissues in the body, so we want to protect it. Stratified columnar. Excuse me. Sorry, it's been a long time. Now, one of my students said they see babies in a nursery. They see babies in the nursery or babies in the where babies are delivered, the obstetrics area of the hospital, and you can see many layers. You can see the top. Here's the lumen, the free surface here, and you can see columnar-shaped cells here, and then one or two more underneath it. See, and so 1, 2, 3, 1, 2, 3. 1, 2, 3, 1, 2. So there are sometimes 2 or 3 cells on top of it here, and always that exists here. But the apical layer, the top, the apex, this part that's near the free surface must be exposed to the air, and that top layer is what we're looking at being long and tall, but more than one layer. It's found in your urethra, that's where you urinate out of, parts of the anus, and your pharynx. And it's again for protection. Now, that is it for the 6 that fit the mold of simple, squamous, cuboid, columnar, stratified, simple, stratified, squamous, cuboid, columnar. Now we're out of that, and we have the 2 oddballs that I like to call them that just are not fitting that thing. And the first one's the transitional epithelium. It looks like the mountains in the old Mario games. And, you look at them, they're kind of rounded here. It looks like the mountains in Mario Brothers where the apical surfaces are curved and round. And there's multiple layers all packed in. And this would be in a bladder that's empty, and there's a full bladder, it stretches out when your bladder fills, and it actually allows its appearance changes as you urinate. It's found in the urinary bladder in the ureters, and if you look at it, since it looks like Mario mountains, and I remember Mario Brothers, the mountains look like that. That's kind of what I see here, and they'd be like but instead, since it's in the bladder, it's beep 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 beep, and it's going to help us remember the curved structure of that. It stretches, it recoils, it stretches, it recoils, it stretches, it recoils. Can do this multiple times, and all these layers of it help us to keep any bad things in the urine from getting back into the body, which is not a good thing. It can end up in the body cavity, and you don't want the things that are urine to get back in. Your body worked very hard to make it and remove things out of the blood to put it into the urine as we got rid of it by the kidneys. So you did a lot of hard work by the kidneys to make your urine. So the last one is pseudostratified ciliated columnar epithelium. What does pseudo mean? False. What does stratified mean? Layered. So it is falsely layered. Pseudostratified. Ciliated. It has cilia. And the cilia, columnar, it's columnar shaped. Look here. Pseudostratified. See how this cell here is, some cells actually can touch the bottom and the top at the same time, but it is falsely stratified. It is not like one layer, another layer, another layer. It's falsely stratified. The pseudostratified and the transitional kind of break the molds. Okay, they don't fit this particular epithelial thing. Now, they're in the nasal cavities and they're in the trachea and the bronchi. Now, I have this meme in my office, hanging on my office wall, and it's an interrupting Kanye meme where Kanye's like, trachea, I know you're helping with breathing and all, and it'll only be a minute, but the nasal cavities have some of the best pseudostratified ciliated columnar epithelium ever. So yeah, but also it's in the vas deferens, ductus deferens or vas deferens. Is there a difference between males and females? Yes, a vas deferens. Um, every now and again, I got to throw something in here funny because I'm telling you guys, I know it's stressful. Now, it functions in protection and secretion. It uses also sometimes the goblet cells to make the mucus. Think of our snot goblet again. And it uses that to catch and trap debris, and then it uses this to swipe and sweep that mucus up out of the throat towards the mouth to cough it up and the things that are trapped in there. It basically acts like a mucus escalator, and the mucus acts like flight paper to trap debris, and then it pulls it up, and you can cough it out and get rid of what you inhaled. It works so well. I remember when I was an undergrad and we were learning about some genetic therapies, my cell biology class, and the professor was telling me, he said one of the things they were trying to do was they were trying to take cystic fibrosis in the cells in the lungs and stuff that was producing this thick mucus because the transport proteins that are found in the glands in the airway are messed up, and you're reclaiming, messing up your reclamation and messing up the secretions, and you end up making this thick mucus that makes it difficult to breathe, becomes fatal for a child. And what they were doing is they were looking at this and they were hoping to use an inhalant to actually implant the DNA into those malfunctioning respiratory cells. Turns out this tissue was so good it prevented that. Now, where we're actually going to stop is after glands. We'll stop right here on slide 27. So the first big thing I need to talk about is, first off, is glandular epithelium. Glands, epithelial cells do make glands, and these glands secrete stuff, secretions. They produce things and secrete it. Now, there's two ways to do this. There's using a duct or not using a duct. Exocrine do have ducts. They do have a duct. Okay? They have ducts. They do produce their stuff, and then they go into a duct. And they produce various secretions like digestive juices, things like that. Endocrine glands, however, do not have ducts. They are ductless. Now, for this, because it is endocrine glands, let's say I have an endocrine gland here, endocrine gland. And over here I've got a cell that would love to hear from endocrine gland. There's only one way to get to it. We have to use our bloodstream. And what endocrine will do is it will use the flow of the bloodstream and it will secrete a material called a hormone. And it actually releases it into the fluid around the tissue called the interstitial fluids. So what we have here is a hormone. And that hormone will then enter the blood. So endocrine secretes endo blood. Endocrine, endo blood. And then the bloodstream will carry the hormone to the cell so the cell can do things about it. So if it's got bunches of these hormones, they'll be secreted, travel through the bloodstream, get to the cells they need to affect. Okay? And that's what they do. So they go endo blood. Endocrine goes endo blood. It secretes in the fluid surrounding the tissues called the interstitial fluid, and then it goes into the bloodstream. And remember, endocrine makes hormones. The endocrine system controls the body using hormones. Now, when those glands secrete, whether it is endocrine or exocrine gland, it can secrete by three modes. There's merocrine mode, which uses vesicles that are emptied out from the gland cells, called exocytosis. This is how the sweat glands, salivary glands work. So usually very watery secretions made by merocrine mode, where the vesicle comes out and dumps its secretions out of the cell. Apicrine secretion, the apex or apical layer of the cell, is shedded and broken down and released. For example, this is how milk is made. Milk is made by breaking down and shedding the apical layer of a cell and releasing it. Holocrine, the whole of the cell dies, releasing its contents, and it actually results in death of that cell. That cell will never reproduce again. It's gone. It has to be made again. Given that, stem cells constantly replace it. And that's actually how sebaceous glands secrete. The oils that your skin's oils are made by that mode. Okay. So things I'm going to ask you. I'm going to ask you, by the way, guys. Then I'll ask you I'm going to ask you the difference between endocrine and exocrine on this exam. Absolutely guaranteed, must know endocrine versus exocrine. Huge, 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 huge. Then you're also going to need to know the three different modes of secretion. I'm going to ask that on the test as well as a multiple choice. Serous glands make very watery things like saliva, for example. Very watery secretions that may have enzymes in it, sweat, things like that. Mucous glands make very thick protective secretions, mucus. Mixed glands make both. Actually, some of your salivary glands are mixed. They do make a mucus, and actually the mixed cells in there, where there's serous cells and mucus cells both, sometimes your serous cells are turned off when you're under stress, and you make a very thick, more mucousy saliva because of that. Now, the last thing I want to talk about is the types of glands. There's two ways to describe them based on the structure of it. If it is one cell or multiple cells. If it's a single cell gland, it's called unicellular. The example of that's a mucus cell or a goblet cell that's the only one. It's exocrine. It makes the mucus. It's actually found in your intestinal tract, your tracheal lining, amongst other epithelium that produce mucus. And sometimes your mucus is really thick, and these glands, there is some malfunctions in the proteins that are acting to adjust it and readjust the secretions, absorb and secrete things properly to adjust the secretion. And when this is malfunctioned, that mutation causes a disease called cystic fibrosis, and we thought we could fix it. One of the things cystic fibrosis patients will also mess with is their sweat glands don't reabsorb much of the sodium and chloride out of their sweat, making their sweat extra salty. And used to be one of the tests for this before genetic testing is they would lick the patient and see if they tested extra salty. So you have a doctor licking a kid going, yep, they're salty. They probably have cystic fibrosis. Cystic fibrosis is still going to be very tough to fight. I think now the best way probably to do this is when it's an embryo is to use CRISPR-Cas9 and edit the genome before birth, before even the lungs develop and things like that, is go in the area where the lung buds are going to come out and edit those genes. You could potentially just edit the whole thing very early on, blastocyst, potentially. I don't know how early you would be able to edit it, honestly, but I think there's where Cas9 CRISPR can come in and help us adjust that. Anyway, guys, so here's an image showing you simple glands. Now, unicellular, simple is a multicellular gland with one duct. Compound glands have multiple branching ducts. Now, that means it's both multicellular glands. There are more than one cell. Here we have simple glands, multiple cells, one duct. Whether it's curved, there's still one duct here. One duct, one duct, one duct. But here, multiple branching ducts. And you're like, well, how is that not multiple branching ducts? They are a branched tubular gland, but still simple because this is the only part that actually the duct. How we do define that histologically is also has to do with the proteins present. There is a assay you can look at where the proteins are for the duct that actually causes things to be adjusted, for example. So you're not going to have to identify pictures of that. Now, so on this, remember the pictures of epithelia, you're going to be looking at this. And if you printed out the instructions I told you guys to use, you can help identify everything, guys. So this concludes our video. My voice is really starting to fail now. This is my 4th recording of the day, so I'm pretty tired. So, guys, thank you so much. I hope you found it useful to see and be guided online. Thank you so much, and we'll see you in the next one. Thank you.